Cut off its leg, it grows back in weeks. Damage its heart, it repairs itself completely. Scientists have injected human cancer cells into it. The axolotl destroyed them. We don't fully understand how. This is not science fiction. This is a 6-inch amphibian that lives in a single lake on the outskirts of Mexico City, and it may hold the key to regenerating human organs. In the southern edge of one of the world's most crowded cities, there's a system of ancient canals left over from the Aztec Empire. They're called Xochimilco, and hidden in the murky water beneath the tourist boats and flower-covered barges lives one of the strangest animals on Earth. The axolotl, Ambystoma mexicanum. It looks like something drawn by a child who couldn't decide between a fish and a lizard. External gills splayed from its head like a crown, stubby legs, a wide permanent smile. Indigenous to this lake system alone. Found nowhere else in the wild on the planet. To the Aztecs, it was sacred. Named after Xolotl, the dog-headed god of lightning and death, who, according to myth, transformed himself into a salamander to avoid being sacrificed. They ate it. They used it as medicine. They believed it possessed supernatural properties. They were not entirely wrong. The axolotl belongs to a group called salamanders, but it does something almost no other salamander does. It never grows up. Most amphibians go through metamorphosis, tadpole to frog, larva to adult. The axolotl skips this entirely. It reaches sexual maturity while still in its juvenile, larval form. It keeps its feathery external gills. It keeps its fins. It stays aquatic. It remains biologically a permanent child. Scientists have a word for this. Neoteny, retained juvenile traits into adulthood. You can force an axolotl to metamorphose, inject it with iodine or certain hormones, and it will transform, losing its gills, developing rougher skin, becoming more like a conventional salamander. But it doesn't do this naturally, and when scientists force it, the animal becomes sickly. It lives shorter. It is fundamentally built to stay young. But neoteny is not what makes the axolotl famous in laboratories around the world. What makes it famous is what it can do when it gets hurt. In labs across the United States, Europe, and Japan, scientists have spent decades cutting off axolotl limbs, not out of cruelty, out of disbelief. Within hours of losing a leg, the axolotl's wound closes. Not with scar tissue. Humans close wounds with scar tissue, stiff fibrous collagen that patches the hole but cannot restore function. The axolotl does something different. Cells at the wound site de-differentiate. They strip away their identity. A muscle cell stops being a muscle cell. A bone cell stops being a bone cell. They revert backwards into something resembling stem cells. This cluster of reverted cells is called a blastema. It forms like a bubble under the wound site, a mass of genetic potential with no fixed purpose yet. And then, guided by signals scientists are still mapping, it begins to rebuild. In the correct order, with the correct proportions, with full functionality: bone, nerve, blood vessel, muscle, skin. Not a rough approximation, the real thing. And it's not just limbs. The axolotl can regenerate its spinal cord, sever it, and neurons regrow across the gap. Paralyze it, and it recovers. It can regenerate portions of its brain, damage the olfactory bulb, the structure responsible for smell, and new neurons grow in to replace what was lost. It can regenerate its heart. Researchers have removed up to a quarter of the axolotl's cardiac tissue. The heart does not scar. It does not weaken. It rebuilds fully. The muscle regrows. The electrical signaling reestablishes. The organ functions as if nothing happened. In humans, a heart attack leaves a scar. That scar never heals. It stiffens the heart wall, weakens the pump, and over years leads to heart failure. In the axolotl, the cardiac equivalent of a heart attack simply fixes itself. The question that has obsessed scientists for over a century is simple. How? In 2018, a team of researchers published something extraordinary. They had sequenced the axolotl genome. It is the largest genome ever fully sequenced, 10 times larger than the human genome. 32 billion base pairs. A library so massive that for years the computers of the era couldn't even process it. Inside that genome, scientists found genes that humans also have. Genes associated with wound response, genes associated with stem cell activity, genes that in humans are switched off after birth, silenced during development, but in the axolotl remain permanently, quietly active. It appears the axolotl did not evolve new powers. It kept old ones. Early vertebrates, the ancient ancestors we all share, were likely capable of significant regeneration. Over hundreds of millions of years of evolution, mammals lost this capacity. The trade-off may have involved the immune system. A highly reactive immune system that attacks wounds aggressively, which is essential for fighting infection, may also be what prevents clean tissue regeneration. Scar tissue is, in part, the immune system's emergency patch job. The axolotl's immune system responds differently. It does not flood the wound with the same inflammatory signals. It allows the blastema to form. It permits regrowth without triggering a full immune assault on the rebuilding tissue. Researchers have identified specific molecules, proteins called TGF-beta, molecules called macrophages, signaling pathways with names like WNT and Notch, that appear to coordinate the process. Each one is a switch. Flip the wrong one, and regrowth fails. Flip them in the right order, at the right time, in the right tissue, and something almost miraculous occurs. The axolotl somehow runs this program perfectly every time without instruction. And there's one more thing that stunned researchers: position memory. When an axolotl regrows a limb, it regrows the correct limb in the correct orientation with the correct number of digits. If you rotate the wound site experimentally, the limb grows back rotated. The cells remember where they are in the body. They know what belongs where. Scientists call this positional identity. It is encoded in the cells themselves, not in the brain, not in the nervous system, in the cells, and we do not yet know exactly how. In the 1960s, researchers at the University of Buffalo did something unusual. They transplanted cancer cells into axolotls, human cancer cells, aggressive ones. In almost any other vertebrate, those cells would proliferate. They would invade. They would form tumors. In the axolotl, something different happened. The cancer cells did not spread. In many cases, they were contained. In some experiments, the axolotl cells appeared to reprogram the cancer cells, reverting them toward a more normal developmental state, not destroying them outright, but converting them. This is not fully understood, but it connects to something deeper about the axolotl's biology. Cancer at its core is a failure of cellular identity, cells that lose their normal programming and begin dividing without restraint. The axolotl, which routinely induces its own cells to temporarily lose identity during regeneration, appears to have powerful molecular machinery for managing that process, for keeping dedifferentiation controlled, for ensuring that cells that let go of their identity eventually become something useful rather than something destructive. Whether this machinery could be harnessed for cancer treatment is an open question. Researchers are cautious. The leap from axolotl biology to human oncology is enormous. But the observation that this animal routinely handles dedifferentiated cells without developing tumors is too significant to ignore. Some scientists have begun studying the specific proteins the axolotl uses to maintain blastema control. Others are looking at whether those proteins could suppress tumor formation in mammalian tissue. The axolotl does not give easy answers, but it keeps asking better questions. Every year, millions of people suffer injuries from which they cannot recover. Spinal cord injuries that leave patients paralyzed for life. Heart attacks that permanently scar cardiac muscle. Strokes that destroy irreplaceable neurons. Amputations where the limb is simply gone. Modern medicine is extraordinary at keeping people alive. It is far less extraordinary at restoration. At the cellular level, we patch, we bypass, we compensate, we almost never rebuild. What researchers want, what the axolotl might eventually teach us, is how to make the human body rebuild. One of the most promising avenues is stem cell therapy combined with molecular signaling. The idea, if we can deliver the right signals to a wound site, the same molecular sequence the axolotl uses to activate its blastema, we might be able to coax human cells to temporarily de-differentiate, to form a blastema equivalent, to regrow. Experiments in mice have already shown partial success. Researchers have activated WNT signaling in mouse cardiac tissue and seen measurable improvements in regeneration after injury. Not full axolotl-level regrowth, nothing close, but statistically significant improvement over normal mammalian response. Others are working on spinal cord applications. The axolotl spinal cord regeneration relies on a specific population of glial cells, non-neuronal cells that form a bridge structure across the injury site, allowing neurons to grow through. Identifying the human equivalent of these cells and figuring out how to activate them is a major focus of current research. And in the field of organ regeneration, axolotl research has fed directly into the science of organoids, miniature lab-grown organ structures that recapitulate the architecture and function of real tissue. Understanding how the axolotl coordinates complex multi-tissue regrowth is informing how scientists think about growing replacement organs entirely outside the body. The timeline is long. We are not on the verge of human limb regrowth. Scientists are careful to say this. The axolotl has 32 billion base pairs of genetic machinery running a system that evolved over hundreds of millions of years. We cannot simply copy and paste. But the principles it has revealed, that mammalian cells retain latent regenerative capacity, that the immune response can be modulated to permit rather than prevent regrowth, that positional identity is encoded at the cellular level. These are insights that didn't exist 30 years ago, and they came from a 6-inch creature in a Mexican lake. Here's the part that scientists don't like to talk about. The axolotl is critically endangered. When the first surveys were conducted in 1998, researchers counted around 6,000 axolotls per square kilometer in Xochimilco. By 2014, that number had dropped to 36, a reduction of more than 99%. The reasons are layered. Mexico City's expansion has encroached on and drained the canal system. Agricultural runoff and urban waste have contaminated the water. And in the mid-20th century, the Mexican government introduced tilapia and carp to Xochimilco as food sources. These invasive fish eat axolotl eggs and juveniles. The animal that may hold answers to human spinal injury, heart disease, and cancer, the animal that survived unchanged through ice ages, through the extinction of the dinosaurs, through 50 million years of geological upheaval, is being killed by pollution and invasive fish in a city of 20 million people. There are conservation efforts. Mexican universities maintain breeding populations. Xochimilco has designated axolotl sanctuaries where invasive fish are controlled. International research institutions maintain genetic archives, but the wild population remains on the edge. We have sent rovers to Mars. We have mapped distant galaxies. We have split the atom and sequenced the human genome. And yet, in a shallow lake on the outskirts of Mexico City, there is an animal doing something we cannot do, cannot replicate, and cannot yet explain. Growing back what was lost, the axolotl doesn't know it's remarkable. It doesn't know that researchers in Vienna and Boston and Osaka are studying its genome, recreating its molecular signals, hoping to coax human hearts into doing what its heart does naturally. It just heals. Perhaps that's what makes it so important. It is not a metaphor. It is not a symbol. It is a biological fact, stubborn, persistent, and extraordinary, swimming in a disappearing lake, holding secrets worth more than we yet know.